Okay. Today, then we will start with DNA fingerprinting, section 3 in chapter 9. DNA fingerprinting is a technique used to identify people at the molecular level. Okay, we said that a couple of decades ago, the lines and swirls of someone's fingerprint were a detective's best hope for identifying a person. Okay, they were using these lines to identify a person. But now investigators gather biological samples and analyze DNA for another kind of evidence, which is DNA fingerprinting. So DNA fingerprinting become now used most because it's a kind of more evidence. Okay, يعني they study the print of DNA at molecular level. At a molecular level means at the size of DNA, which is a representation of parts of individual DNA that can be used to identify a person at a molecular level. يعني fingerprinting or these lines were used to identify persons, but this happened before a couple of decades ago. Now, to give more evidence, they make fingerprinting for the DNA itself, also to identify a people at molecular level, at a level of molecule, very small level, which is what is the molecule? What is the molecule here? The DNA. DNA is a molecule, a polymer, which made of monomers and nucleotides. Now, fingerprinting is a type of restriction map. As we learned before, we need to cut the DNA and use the gel electrophoresis to separate the fragments of DNA, and this will give you a restriction map, okay, to show how the fragments are traveled through the gel electrophoresis. DNA fingerprints based on parts of individual DNA that can be used for identification. This is very, very important. And you should keep in your mind that the fingerprint based on what? Which parts of DNA? It's based on non-coding regions of DNA. Okay? And we said that non-coding regions of DNA have repeating DNA sequences. And the number of these repeats differs between people. So, DNA fingerprint based on parts of DNA which are non-coding regions. When non-coding regions may be the same between several people. What differs in the non-coding regions, not the non-coding regions itself, the repeats of the non-coding regions. Okay, DNA sequences. Okay, so as you see here, person A and B, after they make electrophoresis for the DNA, they make map, restriction map, and it shows here that each person have the non-coding regions, but the repeats are different among them. Any question till here, guys? So, why we use DNA fingerprinting? To identify what? To identify people. To identify people. And this is used a lot for to identify some people if they are relatives, if they are from the same family, if this is a daughter for this father, if this is a son for this mother, to identify people, okay? It compares at least 5 regions of the genome. Usually, to make sure or to give more evidence in the general fingerprinting is right, you should study 5 bands at least. Saddle map, restriction map, after we put the DNA in the electrophoresis. طبعا first DNA will be cut by restriction enzymes. Fragments of DNA will be added to the electrophoresis. Then they will start to move according to their size. Okay. هلا to study for a to compare يعني we make fingerprinting DNA to compare between people fingerprinting to see if they are relative or what do they share to identify them. So you have to study 5 5 regions at least. Okay, 5 regions at least. So here compares at least 5 regions of the genome. Okay, all right. So here main purpose for identification عشان نتعرف على حدا معين من عائلة معينة. Okay. أو ابن فلان ابن بنته ابن بنته. You have to identify people. تمام. This is for fingerprinting. Now let's go down to the next part, which is genetic engineering. Genetic engineering, we said that genetic engineering, just 1 minute, I want to share it with you. DNA sequences of organisms can be changed. Genetic engineering is a technique used to change in DNA sequences. Why? What is the purpose to change DNA sequences? Yes, anyone know? Anyone knows?
Wait, what's the how to change the DNA is what? See the sequence?
Why genetic engineering is beneficial? How is it beneficial in changing DNA sequences of organisms? Yes, Tasneem.
Is it like maybe because we do genetic engineering to give an organism a specific trait that they need or that they think that's best for the organism?
Yes, to change some trait, to change some disorder. Okay. Genetic engineering is something that, or a technique that is used to change in the DNA sequence in an organism. And we said it was an example like when you change the genetic sequence in the watermelon. The watermelon usually have seeds in it. They make genetic engineering changing in the DNA of the watermelon to produce watermelon without seeds. Of course, we have many types of genetic engineering to give the organism better traits to prevent them from some genetic disorders. فبنعمل عنا changing في DNA sequence. We said here إنه before we go to the genetic engineering, I would like to remind you with the cloning. Cloning is also a technique which is used to produce genetically identical copy of gene or of an organism. And the cloning, we said it happens naturally in the environment by bacteria. It clone itself, make exact copy by binary fission, the division of bacteria. Some plants, they divide from the root by cloning. And some animals like yeast, budding yeast, hydra, with sea star, they reproduce by cloning by themselves. This is naturally happen. But in humans and in mammals in general, they can be cloned through artificial process, through a process called nuclear transfer, in which they take a nucleus. A nucleus is removed from an egg. Okay, the nucleus of the cell from the animal to be cloned is implanted in the egg. They implant in this egg cell the nucleus which they need to clone. Okay, why they put it in an egg? Because the egg has the ability to divide. Then DNA from the body is extracted to be used and the rest is discarded. Okay, the rest is discarded. And what they will do, they will take this one, which is the egg with the DNA of the animal to be cloned and implanted inside the body, implanted in or transplanted into another female. هلا the offspring that will be produced will be similar to the one before. Okay, هلا cloning has advantages and disadvantages. Advantages انه some يعني they can use the cloning for organ transplantation into humans. يعني some people they have problems in their liver and function of the kidney, and kidney doesn't make their function properly. They need new organs, but they use the cloning to transplantation of organs into humans. And because there are some endangered species, they save them by cloning. But the cloning, on the other hand, has disadvantages like low success rate, and it is imperfect and less healthy than original animal because there is a premature aging and old DNA. You are taking the DNA of an old or premature, yani old animal will be cloned. So the produced or the generated animal, a new animal, will have premature aging and old DNA. And this also will decrease biodiversity. Sometimes also clone may not look like and behave like the original. Okay, because now we said there are many factors that affect the phenotype, not only the genotype, also the environment affects the expression of genes. Okay, now هذا الكلونينج help the genetic researchers to use the genetic engineering, okay, and to copy some segments of DNA. يعني احنا بنعرف انه الكلونينج هو copying of the same or the same genes or the same organism. They use this idea by cloning or copying of some genes, not all the DNA, genes in the DNA segment, okay, and change these genes or insert new genes for one organism to change in their traits or their disorders. They rely also at the same, as we said at the beginning, in all these biotechnological techniques, they relies on cutting DNA. فهون the new genes can be added to an organism DNA. تمام؟ Genetic engineering involves changing an organism DNA to give it a new trait. هاي هي الفكرة. The genetic engineering you make altering يعني بتعطيني organism that have altered genome. الأورجانيزم اللي بيطلع عندي على genetic engineering صار عنده alteration في الجينوم تبعه، يعني الجينات تبعته تغيرت. Or some change, not all of it, some change happened. Okay? Genetic engineering based on the use of recombinant DNA. Recombinant DNA هذا contains genes from more than one organism. وقلنا هذا recombinant DNA how they make it. They take plasmid bacterial DNA from bacteria. Okay, and they add the طبعا بيستعملوا برضه restriction enzyme. They cut where they want to add the gene, and they add the foreign DNA or the foreign gene from the DNA to this plasmid bacteria. Okay. Okay, هذا no problem. شايفين هون. This is the plasmid. بيستعملوا restriction enzyme to cut the DNA, and then we add the wanted gene. This will change in the trait. وبعدين this will produce new bacteria plasmid with new genes يعني new genome. فأي genetic engineered organism معناته عنده his genome is altered تغير اختلف not as the original. Okay, sometimes you alter one gene or more. Okay, a transgenic organism has one or more genes from another organism inserted into its genome. هذا هو transgenic organism. أحيانا بيكون عندي transgenic plant, sometimes بيكون عندي transgenic animal زي watermelon. When the watermelon was changing to one without the seeds, هذا اسمه transgenic watermelon because it has altered its genome. Okay. طيب. Okay. طيب. The plasmid and foreign gene are bonded together to form هذا اسمه when you add the new gene to the plasmid, this will become a recombinant DNA. Okay. طيب. هلا after they inserted gene inserted to the plasmid بحطوا البلازميد في البكتيريا والبكتيريا will express the gene. Okay. They may have one or more genes as we said. We can use this type of genetic engineering to produce insulin. The plasmid are inserted into bacteria. بياخدوا gene من a human or animal which make insulin. They add it to the plasmid of the bacteria. Then the transgenic bacteria start making human insulin, and it will be collected and it will be sold as treatment for people with diabetes. قلنا في example على transgenic organisms زي transgenic plants. اوكي. وفي عندي transgenic animals. طبعا بالtransgenic plants it's easier to change the gene in a plant. It is inserted into the plasmid, then the plasmid is inserted to the bacteria. Then they let the bacteria to infect the plant, and the new gene become part of the plant's DNA. And it will be expressed, of course. Okay, and here transgenic plants, like why they make transgenic plants by genetic engineering to increase food production, to save endangered species, to make plants that resist pesticides. وفي عندي something called Bt toxin, is a bacterial gene that is lethal for insects. They change in the gene of the plant to have this Bt toxin, this gene, and this gene is very useful because it gives the bacterial the ability to let the insect away from it, because this is lethal, okay, for the insect. Okay, تمام. So here, with transgenic animals, we said that it's harder to produce transgenic animals than the plants, okay, because they are more resistant to genetic manipulation. And it have a more complicated procedure, because transgenic animal, the researcher must first get a fertilized egg, then they add the DNA into the nucleus, and the egg is implanted back to the female. يعني عمليتها معقدة أكثر. ياخذوا fertilized egg, after the sperm and egg are joined together بصير عندي fertilized egg, then they will add the foreign DNA into the nucleus. بعدين الايج هاي will implant it back into the female, يطلعوها out, بعدين they give it infant. It's harder to make. Okay. Example of transgenic animals, they high yield milk producing cows. They produce some cows produce milk with similar properties as a human breast milk. Okay. وفي عندي كمان researchers have developed GM dairy, a cattle. GM means genetically modified. واحنا قلنا genetically modified يعني الجينوم تبعها was altered, it changed because new genes from the DNA was added or inserted in the nucleus or the DNA of the organism. وفي عندي كمان transgenic animals like fast growth salmon. Okay, any question till here, guys?
No, teacher.
Clear?
Yes.
Okay, وفي كمان عندي برضه transgenic animals used to study diseases. يعني زي عندي sometimes they use المايس for study many diseases, للكانسر مثلا أو للobesity. ف they call these animals that used for study oncomouse. هذا الماوس used to study some disorders بنسموه oncomouse. They want to study the development of disease. وحكينا function مثلا example على واحد من هدول الستاديز. they study the obesity. okay. شو بيعملوا look out for gene، بوقفوا جين معين عنده. okay. and this gene will become not functional. okay. and this gene مثلا study obesity بهذا الماوس، فبيعملوا look out, they stop the functioning of this gene اللي هو مسؤول عن الobesity اسمه leptin. هذا control food intake. فبوقفوا عمل هذا الجين and they will see the effect of this stop gene on the obesity. يعني they have many ways. هلا أهم شيء تعرفوا لي هون عن genetic engineering إنه أي organism that genetically engineered or genetically modified means that their genome was altered. يعني صار عندي change بالدي ان اي by inserting a new gene to the genome of this organism. And they use recombinant DNA by taking the plasmid من bacteria. بيعملوا cutting للplasmid. قلنا الplasmid هي DNA part موجود في البكتيريا. They cut the part they don't want and they add the part that they want by sticking them together. بصير عنده اسم هذا recombinant DNA and they will insert it back to the organism. فهيك بصير عندي أنا الترانسجينيك organism أو genetic engineered أو genetically modified. شوفوا الثلاثة هدول نفس المعنى. لما أقول أنا transgenic organism the same meaning as genetic engineered organism, the same meaning as genetically modified. نفس المعنى إنهم all of these have altered their genome. يعني الجينات بتاعتها تغيرت. Clear guys?
Wait, can you repeat it, Miss?
Yes, please.
We said if you say transgenic organism or genetically modified organism or genetically engineered organism, they have the same meaning. In their genome or their genes, maybe one or more genes were altered, changed. Yeah.
Okay.
Any questions? So, let's go to the next part now. اللي هو genomics and bioinformatics. هذا genomics و bioinformatics I just want you to know few things about it. يعني ما بدي details. بدي تعرفوا إنه الجينومكس شو معناها والbioinformatics شو معناها. Here we said entire genomes are sequenced, studied and compared. الانتير جينوم اللي هم الجينات اللي هم النيوكليوتايد في الدي ان اي. هدول هم الجينات السكشنز in the DNA. كل سكشن بيعطيك جين و all the gene هو الجينوم. اوكي هلا here genomics involves the study of genes. بس نعرف شو يعني genomics. هو study of genes والجين functions والانتير جينوم انه كل الجينوم. Genomic is a study of genomes which include the sequences of genome. ايش هو السيكونس قصدنا احنا فيه؟ النيوكليوتايدز هذه are sequence or comparisons of genome within the and across species. If you want to compare between the genes of مثلا two species, the human ومثلا as we said before chimpanzee or two species of birds, you want to compare between the genomes. This is genomics. Okay? This is the first thing you have to know. And we also talk here about something in the genome. Genome, when you hear this word, genome means most DNA, most of it. Okay? And they found here that مثلا الجينوم for different species are different. يعني عندي الإي كولاي هاي الإي كولاي type of bacteria عندها 4.6 million bases. تخيلوا الجينوم هم السيكونسز للبيزز اللي هم النيوكليوتايد. فالإي كولاي عندها 4.6 millions of bases. If you compare it to humans, the humans 3,000 millions of bases. While we have other species more than us, the lungfish, زي vanilla plant that you use in the cake, هاي vanilla seeds, they have more genome than us, while they are smaller. فما دخل. إذا هو الجينومكس هو study of the genome of the DNA sequences والfunctioning. هلا في عندي another thing هون كمان used. في عندي project اللي هو الجينوم project. هذا sequenced all of the DNA based parts of the human chromosome. Okay, and يعني لسه still working to identify and map the human gene. Okay, now there is a technique or method used to as a computer database to organize and analyze biological data. يعني when you collect any biological data about the sequences of the genome or etc., then there is a computer database called bioinformatics. This is very useful to organize and analyze your data. The microarrays used to study the expression of many genes. Proteomics is the study and comparison of proteins. Any question? Let's go to the last section in this chapter, genetic screening and gene therapy. من اسمه gene therapy، شو يعني therapy؟ What was the meaning of therapy? Curing.
معالجة زي
Yes, بالضبط curing. يعني when you have some disease مثلا you have headache, you take Panadol or aspirin. This is a cure for the headache. صح؟ هلا في عندي some disorders they can be cured by gene therapy. طيب فعشان نعمل الجين therapy first they make genetic screening. how بالضبط لما نروح على الدكتور and الدكتور will diagnose their symptoms. You see what ask you about the symptoms, what do you feel, what if you have any pain. The doctor making their diagnosis عم بيحدد شو المرض. نفس الشيء هون. Genetic screening is to assess أهم شيء assess risk for different disorders. بيشوف يحدد هل في ممكن يطلع عندي disorder ولا لا. عم بعمل screening على الجينات. After this they will see if they can cure this defect. إذا كان عندي defect معين أو disorder معين بقدر يستعملوا gene therapy. طبعا not in all cases, not in all diseases. لسه هذا الموضوع تحت الدراسة and بيعمل عليها researching كتير. بس هي هاي ببساطة. Genetics provides a basis for a new medical treatment. إحنا كثير عندنا medical treatments for many diseases. بس هلا حتى الجينات provide basis for a new medical treatment. حنحكي عنها. أوكي؟ هلا إحنا sometimes زي ما قلنا في عندي sometimes people هم they are not diabetic ما عندهم مرض السكر but they are carrier for genetic disorder. فالجينيتك سكرينينج detect genetic disorders. نعمل genetic screening بيعمل assessing الريسك قديش الريسك عندي انه الاولاد يطلع عندهم diabetes. فبيعمل screening او assessing the risk. اوكي؟ اذا detect genetic disorders بالذات اللي بيكون عندهم carriers for genetic disorders. They don't show the phenotype بس هم carrier. اذا هاي تعريفه genetic screening is the process of testing DNA to determine person's risk of having passing disorders than offspring. Okay? Genetic screening often involves pedigrees and family histories as well as DNA. يعني كثير تستخدم برضه معها genetic screening البيديجري والفاميلي histories والDNA testing. هلا برضه they can detect genetic disorders عن طريق comparing الباند. نفس الشيء بيعملوا restriction انت بيعملوا cutting بيطلع عندي باند بيعملوا map restriction map they compare it. وشو sorry
بس بدي احكي لك you are not sharing a screen. احنا مش شايفين شيء.
really
yes.
من امتى؟
من السكشن
from the beginning. Okay, sorry guys. I thought that I shared it. Okay, I will repeat again. We said in the genetic exam screening with gene therapy. This is the definition of genetic screening, process of testing DNA to determine the risk of having passing disorder. لأنه we said some people they are carriers for certain disorders and they didn't show the phenotype. So to detect if they can pass this risk, or if there is risk to pass this disorder or not by genetic screening. And it also uses or involves the pedigree and the family histories and DNA testing. So it can detect genetic disorders, determine risk of having or passing on a genetic disorder, used to detect specific genes or proteins, can detect some genes related to an increased risk of cancer. كتير ناس بيكون عندهم risk or high risk of cancer. They can detect it by genetic screening. Some genes known to cause genetic disorders. زي عندي Dutchens muscular dystrophy. Okay? They use the same procedure. بياخدوا DNA, بيعملوا cutting, بيعملوا electrophoresis, then they compare it to the band for different persons. Okay. طبعا not all the genetic disorders can be screened. بس احنا قلنا under studying. هلا if they detect disorder, some cases or some disorders can be cured by gene therapy. Okay. So how they cure it? Gene therapy replaces defective or missing gene or adds new genes to treat a disease. بيعملوا adding كيف عند الجينيتك engineered animals والبلانت عملنا adding لنيو DNA gene. نفس الشيء هون بياخذوا ال take away or add some new genes to treat a disease. طبعا not all diseases can be treated in this way. First, they get the new gene into the correct cell of the patient body. Once the body gene has to become part of the cell DNA, one method of gene therapy that scientists have tried to take a sample of bone marrow. يعني one way حطوا بالكم هذا الإكزامبل إنه they take sample من البون marrow نخاع العظم, okay, stem cells, and infect them with a virus. بحطوا جواته virus that has been genetically engineered. طبعا هذا الفيروس before they put it في هدول السيلز هذا الفيروس it changed the DNA genetically engineered مغيرين في الدي ان اي تبعه with the new gene اللي بدهم اياه. Then بحطوا الفيروس بهذا الستم cell, then they will put back this stem cell into the patient bone. The stem cells هدول هم cells that differentiated to different types of cells. زي بصير منهم بعضهم red blood cells منهم white blood cells and they make more blood cells with the gene. يعني كمان مرة بالجينيتك الجين ثيرابي شو بياخذوا دائما عادة بياخذوا stem cells يعني cells عندها القدرة انها to differentiate. شو يعني differentiate؟ يعني يصير لها division ل different types of cells. فالبون مارو نخاع العظم هذا فيه stem cells. هدول الstem cells can divide to white cells و red blood cells و platelets. اوكي طيب شو بيعملوا؟ بدهم يغيروا في مشكلة معينة ب this person. بياخذوا هدول الstem cells they put inside them virus. هذا الفيروس بيكون genetically engineered مغيرين بالجينات تاعونه. They put it inside the stem cells and they put the stem cells back. هلا الستم هذا الفيروس have the gene. ف patient will have the gene in their cells. لأن الفيروس رح يصير له replication رح يتكاثر جوا السيل and they will divide with the new cells blood cells with the new gene. هلا هي ما راح اجيب لكم اياها انا الطريقه. I will not bring this يعني بهذا الديتيلز بس انا بدي تفهموا. شايفين هذا الفيكتور حطوه جوا السيل. السيل جواته الدي ان اي هيو. الدي ان اي بتاعتها كلها بلو. في عنده هذا الفيروس نيو جين was added to it فهو اصلا engineered فيروس. بحطوا الجين اللي بدهم اياه and they will add it inside the stem cell والstem cell will divide with the new gene. في سؤال؟ هذا مش مهم. في سؤال؟ No question?
Wait, Miss, can you repeat the bone marrow thing?
البون marrow they take a virus. بهذا الفيروس it has a DNA. They change the DNA of the virus by adding the gene they want in the virus. Then they will infect the stem cell from the bone marrow with the virus and put it back in the body of the organism. This stem cell will start to divide to new cells with a new gene. So the gene will be expressed and the new cells will have this gene in them. Okay?
Okay.
Tamam. Now let's go and start like a very small introduction in the new chapter اللي هو chapter 13 اللي هو ecology. Ecology, these lessons actually are very simple, but you have to concentrate while you are answering the questions in the exam. The first, whole chapter 13 is about ecology. The first section, ecologists study relationships. The most important thing you have to know here, what is ecology? The study of the relationships among organisms and their environment. And you also should know that ecologists study environments at different levels of organization, which are, what are the levels of organization? With any relationships start with the organism. Levels of organization start with an individual, which is organism, and this organism, group of it will make a population of the same species will make population. many populations of different species will make community.
مس
نعم.
سوري لأنه بضل أفوت وأطلع بس my internet connection is bad.
Okay, no problem.
Thank you.
إذا levels of organization will start from إذا قلنا من smallest to biggest organism, population, community, ecosystem which include living and non-living things اللي هم communities plus the environment around it abiotic factors, biotic and abiotic factors. Then after the ecosystem we come to the biome. Okay? إذا لو قلنا بالعكس طيب levels of organization from the biggest to smallest start from what? Who knows? If you want to arrange the levels of organization opposite from largest level to the smallest level. مين أكبر إشي؟ Biome, then ecosystem, then community, population, organism. And the opposite is from smallest to biggest اللي هو organism, population, community, ecosystem, biome. هذول هم levels of organization. هذول لازم نعرفهم levels of organization. وطبعا they use different methods. They use observation, they use experiment, and they use modeling to study the relationships between organisms. So we will stop here لأنه time of the class finished, and I will meet you at 1:30. Please tell your friends that these classes are very important, okay?